discovered, nearly all Staph aureus would respond to penicillin. So the number of resistant organisms was very, very low. But as soon as penicillin became introduced, the fraction of Staph aureus that was resistant grew very rapidly in hospitals, but even in the general community caught up with it. So today, nearly all Staph aureus is resistant to penicillin. Now, there are other antibiotics that Staph aureus is sensitive to, but in fact, there are now multi-drug resistant Staph aureus that responds to only one antibiotic, which is vancomycin. And there are actually reports of vancomycin resistant Staph aureus, although not in combination with multi-drug resistant Staph aureus. So it's clear that if these things combine, we could have a Staph aureus that doesn't respond to any known antibiotic. And so we really need development of new antibiotics. And you can see that in England, there are a number of deaths in hospitals due to these multi-drug resistant Staph aureus. And this is John Reed, who was the health secretary at the time in 2004, asking hospitals to take action about infection rates. So the whole idea of antibiotics in the modern era, of course, traditional medicines always had some thing that they would use to treat infections with, but without actually knowing about bacteria and so on. And the first systematic scientific effort to try and develop something that would target bacteria was by Paul Ehrlich, who observed that if you stained bacteria and looked at them under the microscope, there were certain dyes that would be preferentially taken up by bacteria, but not taken up by a human cell, the lymphocyte cell. And so this led us to think that maybe if you had a dye that was toxic and bound specifically to bacteria, it would then have a magic bullet that would only kill bacteria and not harm organisms. And Paul Ehrlich was not, he had the idea, but he wasn't quite so successful in producing a useful antibiotic. And that was first done by this man, Gerhard Domas, whose contribution was interesting because he worked for the Bayer chemical company, which made a huge number of organic dyes. And he very systematically tried all of these organic dyes for their effectiveness against bacteria. And he came up with this compound called Contosil, which at the time was miraculous because it was the first highly effective drug that you could give to humans, and it was effective against fatal infections. And at the time, streptococcal infections were very, very serious because you could get a cut and it would get infected by streptococcus, and the streptococcus would simply grow along from the region of the cut. And often you would have to amputate an entire limb, or it would be too late and it would become systemic and you would die. And it turned out that soon after he discovered Contosil, he was able to save his own daughter from having her finger amputated. Now, one problem with Contosil was that when people tried to understand how Contosil worked in humans, it turned out that Contosil was broken down into humans, and one of the compounds was a previously known compound called sulfanilamide, so he couldn't patent it. His patent on Contosil was immediately voided because sulfanilamide was already known, so it voided his patent. And there's another unfortunate thing that he had, which was that he was given the Nobel Prize for his discovery of the first sulfa drugs, which was Contosil. But this was during, I think it was about 1940 or something, or 30, no, it's actually in the late 30s. But by that time, the Nobel Peace Prize had gone to a German pacifist who was obviously opposed to the Nazis because of their aggressiveness. And Hitler thereby banned all German scientists from accepting the Nobel Prize. So he was not allowed to go and accept the Nobel Prize because he was in Nazi Germany. After the war, he was invited to a Nobel celebration, prize ceremony, and they gave him his medal and his certificate, but they told him they couldn't give him any money because the money had been returned to the general funds, so which was really quite unfortunate, but he's a great scientist to whom we owe modern antibiotic discoveries. Now, Penicillin and other sulfur drugs were synthetically made. They were made by the chemical industry. And the whole idea of natural antibiotics arose due to penicillin, which was discovered by Alexander Fleming, when he accidentally discovered that a mold spore in a plate in 1929 seemed to give off the compound and prevented bacterial colonies from growing. And so you can see that the colonies are quite large away from the mold spore, but as you get closer, they don't seem to grow. And he correctly surmised that there was a compound secreted by the mold spore, and he called it penicillin because it was the penicillin mold. And penicillin became the first naturally produced antibiotic that was highly effective. It had very low toxicity, and it saved millions of lives actually during the war, towards the end of the war, because a massive effort was made to make large amounts of penicillin. Subsequently, people explored lots of other bacteria, especially soil bacteria. A single genus, Streptomyces, has given us a vast number of different antibiotics. Streptomyces is a genus of soil bacteria. And if you look at all these antibiotics, all of them that were isolated from these various strains of Streptomyces have one thing in common, and that is they all work by blocking protein production in bacteria. So this naturally leads to the question of what are proteins and how are they, why are they important and how are they made? Okay. So I'm going to show you a lot of molecules. So I'm going to show you these molecules in different ways. And so just to tell you how we can show the same thing in different ways, I just want to illustrate that using humans. So on the left, you have a very detailed image of the human being. Okay, that's what we call a very realistic representation. But you can also greatly simplify this and still convey the concept of the person. And that's shown here in terms of cartoons. So the cartoon is very simple compared to the actual person, but nevertheless conveys some essential information, essential features. And if you can simplify this even further by showing the object symbolically. I don't know why I need to see myself. It's not actually something distracting. Okay, so you can still see, you can still show the person as an icon, and if you know what the icon represents, what aspect of the person the icon represents, you immediately know that this icon stands for Clinton or this icon stands for Bush. Okay, Clinton would never be able to decide anything on, at least early in his term, and so he was called a waffle, because the waffle means to not be able to decide. And this comes from a Texan phrase called all hat and no cattle, which suggests that there's not much underneath the hat. And so that was the representation for Bush. So you can similarly represent molecules in very different ways. So this is the molecule DNA, which was discovered by Crick and Watson, the structure. And this is a mechanical or metal model that was drawn to a very large scale. And here they are in this historical photograph. Now, if you were to portray it in a computer and show all of the atoms, then this is what it would look like. Although the atoms are not drawn to their proportional sizes, but at least you can see all of the atoms. But sometimes you want to simplify it even further, and so this is a very standard way of representing DNA by showing these two ribbons wrapped around each other. And you want to show that DNA is composed of 4 types of building blocks, so you can give them 4 different colors, and to show you that A will only fit with a T and G will only fit with a C, you schematically made the joining between these building blocks different, so that you convey the idea of two pegs fitting with each other. Of course, the reality is nothing like this. It's actually hydrogen bonds that are where you have complementary acceptors and donors. But nevertheless, this actually conveys the concept of the architecture of DNA. So when I show you lots of molecules, you need to keep in mind that the representation may be different. So we talked about proteins, and I said that many antibiotics work by blocking protein production. So the question is, what are proteins and why are they important so that blocking them should actually kill bacteria? Well, I'm showing you three different proteins here. One is collagen, whose structure, incidentally, was first discovered in Madras by Penicillin. And you can see that it's a long filamentous protein. This is the protein that makes up our skin and cartilage. This is hemoglobin, which consists of 4 different subunits shown in different colors. And each subunit carries a moiety called a heme, which has an iron atom which bonds to an oxygen molecule. So this is a protein that carries oxygen from our lungs to our blood to go into the tissue. And this is a protein called rhodopsin, which is present in our eyes, and it's a very sensitive photon detector. So you can see proteins carry out a wide variety of functions. In fact, they carry out thousands of functions. And that's why if you stop protein production, effectively none of the essential reactions in the cell will proceed, and so you end up killing the cell. Now, to understand how proteins are made using the information in our genes, okay? So our genes are made of DNA, and as I pointed out, they consist of 4 different types of building blocks, and this is a more chemically accurate representation of this very simplified cartoon. So the backbone of DNA consists of a phosphate, sugar, and phosphate alternating, but each sugar is attached to a different kind of base that is complementary to another type of base. So you have 4 bases, but A will only pair with a C, and C will only pair with a G within the context of the helix, of a double helix of DNA. Now, it's the sequence of these bases that contains information on how to make proteins. And that was a big puzzle in the early days of molecular biology. So here's DNA, and DNA is copied. One of the strands of DNA is copied to make a rather similar molecule as far as information content goes, called RNA, which is a crucial difference that instead of deoxy, instead of just a proton here, you have a hydroxyl group here at the 2' moiety of the sugar. Now you have to go from the language of nucleic acid, which has these 4 types of bases, to the language of proteins. Proteins are long chains of amino acids which fold up into the shapes that I showed you. For instance, in those 3 proteins I showed you, they have very different shapes, but all of them start off as long chains of amino acids that fold up into their particular shape, depending on what the protein is. And so we need to go from this language to this language, and that's what used to be a big puzzle. And the way that's done